Laura Martin, where's your wine?
I'm not going to drink wine. It's here, but
it's happy hour. What do you think we're doing? There you go.
There you go. Yeah.
Cheers. Abel, no hardball questions, okay?
Hey guys, we're live. It's a Monday, 5 p.m. Science Squawk. So I am jumping off this call. I am turning it right over to our host, Abel.
All right. Thanks a lot, everybody. Thank you, Amanda, our executive producer, for, again, hosting another one of these sessions for us as we get used to this this venue, right? My name is Abel Gutierrez. I am the host of Science Squawk. And with me, again, is author extraordinaire, Steve Chappell. And with us today is a very special guest, Dr. Sumit Chanda, who is the Director of Immunity and Pathogenesis Program at let me see if I nail it, the Sanford, Burnham, Prebys Medical Discovery Institute. Did I get it right?
Yes.
All right. Say Sanford Burnham for short, I guess. And with Sumit today is, let's see, who's Laura 1?
Martin-Sancho's Laura 1.
Okay. So we have Dr. Laura Martin-Sancho, who is one of the critical pieces of research here in, or one of the critical researchers here in Sumit's lab. And we have Laura or Laura? Laura as well, and that's the Italian version?
Laura Riva, yes.
Yeah, Laura Riva. And again, thanks guys for spending some of your precious time. I know you guys have been doing this whole media circus with all the news outlets and all the press releases you guys are doing, which I'll get into in a little bit. This is a show to bridge the gap of between scientists and the non-science community, as well as the science community as a whole on the Mesa. So it's really meant to be a conversation. We're trying to highlight the movers and shakers in the area, a little bit on the economy and the economic impact in our city. But really, it's just to have a conversation and help disseminate some of the real facts and the real science that's going on. And hopefully it spreads and helps to diminish some of that misinformation that's out there in the public. So let's just pretend, we're at the Bella Vista Social Club, right? We're all hanging out. And, I guess we'll start with Dr. Chanda or Sumit if I can.
Of course, Abel.
All right. How would you introduce what you do to somebody you meet at the Bella Vista, not knowing their background? Can you?
Sure. Yeah. So there's two time periods in our lab. So one is BC, before Corona, and one is now. So right now it's all Corona all the time. But before that, we were looking at what happens essentially when a virus and our cells meet, right? So the viruses we work on are called RNA viruses, which is coronavirus is an RNA virus. They're fairly compact viruses. They usually encode somewhere between 15 and 30 different proteins. So in contrast, our cells have about 20,000 proteins. So what these viruses really need to do is turn some of our proteins into helpers. So essentially hijacking our own proteins for their own benefit. And so one of the major focuses of the lab is understanding which of those proteins are critical for the virus to usurp and use to promote their replication or their ability to make copies of themselves, okay? And so you can imagine that if you can go after one of those proteins with a drug, you can essentially starve the virus, and that's something that we're doing now with coronavirus. The other aspect that we're looking at is trying to understand how the cell defends against viral infection. So each of our cells have an intrinsic mechanism by which they can sense the virus infecting and then set off alarm bells and essentially trigger a molecular response that is designed to thwart the virus replication. But then you have this arms race going where the virus then comes up with a strategy to thwart the cells and so on and so forth. So understanding that interplay, which is called innate immunity, is the other focus in the lab. And so prior to this, we were working on, so BC, we were working on flu, we were working on dengue, we were working on HIV, a number of other RNA viruses. At this point, the only virus that we are working on in the lab now is SARS-CoV-2. Hopefully with things ramping up a little bit and opening up, we can get back to our day jobs. But I think the two Lauras here are going to continue to be plugging away at trying to find solutions for COVID-19.
That's awesome. So
how's it looking? What's the progress, for the elderly here?
So there's two fronts here, right? One is this social distancing, quarantining. So at least in California, things look good enough by which the governor now is going to start relaxing some of the rules and start getting somewhat back to normal. We shouldn't get complacent. We need more and more testing. That's going to be a key cornerstone for us to be able to return to some semblance of normal life. The other ray of sunshine here was a drug by a company called Gilead up in the Bay Area. Sure. So they have a Ebola drug that they what's called repositioned or moved it from Ebola to test on coronavirus. And the results of those clinical trials have not been put out in peer review publication yet, but initial indications really suggest that we're at, this is a critical first step for treatment. So there were positive outcomes in the number of days people ended up spending in the hospital, and there seems to be some survival benefit. It's not a cure, but it's a treatment. And so if you think back to the days of HIV, the first drug for HIV that came out that was similar was called AZD. This is what I view this as being, right? The first step. Now we have a very advanced cocktail for HIV that essentially makes it a chronic manageable disease. This molecule, remdesivir, is a critical first step. I think work that we're doing, work that others across the globe are doing, are going to be able to add number 2 and number 3, hopefully in time for a likely resurgence in October. And we'll have some treatment options that will really suppress the mortality rates, right, to a point where it is a manageable disease that the way we manage flu will be the way we manage coronavirus. And then, of course, I think the most important thing is going to be the vaccine, right? So people are saying 18 months. To me, that's hugely optimistic. But let's hope that they can pull out all the stops and get that done.
Very cool. Now you mentioned compounds and drugs and treatment. Now for someone that just doesn't really know what's going on left or right, what's the difference between that and an antibody? Would you prefer another, etc.?
No, so there's 2 ways that you can go after a virus. So one is an antibody. An antibody is what our immune systems make to naturally defend against the virus. And so the thought is that if you've gotten infected by corona, you'll have some level of immunity for some amount of time. So this hasn't been definitively shown, but it's the conventional wisdom right now. Is that, and those antibodies essentially are proteins that our immune systems make. They bind the virus and essentially muck it up so it can no longer infect cells, okay? And you can either take those antibodies and make antibody therapeutics, right? So build and engineer proteins that can be injected to help you control the virus. Or use what's called convalescent serum. So you take serum for people who've already been infected. They have these antibodies, and so there are clinical trials going on right now to see if taking serum essentially the antibodies from people who have been infected and see if they're protective or helpful for people who are sick, okay? Small molecules are actually things that go after proteins either in the virus or, as I was explaining before, in our cells that the virus depends on, okay? So they don't bind to the virus and muck it up. They actually are warheads that block typically what's called an enzymatic function. So a protein, a specific protein that functions to do something like replicate the genome of the virus, right? This is what remdesivir does. It targets a protein within coronavirus, a polymerase protein, which blocks the ability of coronavirus to make copies of itself. So it's a very targeted type of approach that goes after the activity of a certain protein, either within the virus or within our cells. Both can be potential arrows in our quiver. And so a number of these strategies are either being developed and evaluated. But the only one right now that has, I would say, unequivocal clinical data to support its utility is remdesivir. And if we can get 2 or 3 more, I think we'll arrive at a cocktail where the virus can't become resistant. So as you know, a lot of viruses, if you throw drugs at it, they can become resistant. Bacteria, happens with antibiotics. It can also happen with viruses. And so typically, if you throw 3 drugs at a virus, it has a much harder time becoming resistant. So this is one of the rationale that people are thinking about combining different antivirals, so we can box the virus into a corner, so we won't see a resistant strain emerge, say, next year, when and if the virus comes back.
Are you saying, though, that you kind of have to get put in the hospital and diagnosed and all of this before you can use remdesivir or some of these others, that in other words, it's not a prophylactic and it's not something that, like, you get tested and you can just take it or something?
Yeah. So there's a couple of issues, right? So right now they're giving remdesivir only to people who have progressed to disease. And it's an IV drug, right? So you have to go into some sort of clinical environment. So they have to hook a line up into your bloodstream to deliver it. So you're not going to be able to take it at home, you know. The other issue right now is that, unfortunately, it's a pretty hard drug to synthesize. So there are not limitless quantities of remdesivir available. And so right now there seems to be enough to go around, but if cases spike up worldwide, there may not be. And so this is another reason why we need additional drugs, so you can drop the dosing of remdesivir with another drug, so there's more to go around. But right now, these clinical trials are designed at looking at treatment. Okay, so if you're sick and if you have symptoms that will either make you likely to go into the ICU or are in the ICU, those are the first people that are getting the drug. And so it's unlikely that unless you have one of the pre-existing conditions, over 65 or some other underlying conditions, you will get remdesivir as a frontline treatment. You'll most likely get supportive therapy, even if you get into the hospital. But that's a moving target. Prophylaxis studies, I don't anticipate will be done anytime soon. Everyone's focused on making sure that the people who are sick aren't dying. Because again, 80% of people who get this virus, mild to almost no symptoms, okay? The key is focusing on the people who are ending up on ventilators, who are in the ICU, and finding ways to move the needle on the mortality rate, right? So to save those people. That's really the thing that everyone's looking at now. As
Good.
Time progresses, I think, people will start looking at prophylaxis, but that's not where the focus is right now.
As somebody a little bit over 65, I would prefer massage therapy, but I guess
Yes. Yeah. We can put that into the clinical trial, see what happens. Yeah.
And then the two Lauras here, are they your bosses? I'm a little confused.
Yes, I work for both of them.
Oh, I see.
So,
are they your postdocs or professors at Sanford? Or?
So they are scientists in the lab, and we can have them introduce themselves. But I can tell you that ever since January, when this hit the radar, these guys have just been heroic in their efforts. They're the ones who go into the biosafety level 3 facility where you have to put on the spacesuit to handle the virus. Really just been a remarkable effort day and night trying to find cures and therapies for the virus. So Laura, why don't you introduce yourself and talk a little bit about the project that's ongoing on the lab. Laura, Riva, why don't you go ahead first?
Okay, I'm Laura Riva, and as Sumit was mentioning, I'm a postdoc in the lab, and since January, we started this project on coronaviruses. So before that, I was working on drug discovery related projects in the lab. So we were trying to find antivirals blocking several positive strand RNA viruses and also negative strand RNA viruses, with a main focus on emergent viruses, including Ebola virus, Zika virus, as well as re-emergent viruses like dengue virus, yellow fever, Japanese encephalitis. But yeah, since January, all the efforts are focused on COVID-19. So it came up, well, of course, there was the outbreak ongoing, and we had the ability of perform drug discovery screens. The problem at the beginning was getting the virus, because in the US, the disease was still not here and the virus wasn't here exactly. So the only place where they had the virus isolated and they were able to use it in culture was actually in China. So Sumit got contact with people in the University of Hong Kong. So we started this collaboration with those people who had the virus in the lab. So in the lab is the lab of Professor Yuan, who is the professor who discovered actually SARS-CoV-1 back in the days. And we started this collaboration. So it was very interesting, I would say, because yes, they had a lot of experience with coronaviruses, but they had no experience at all with drug discovery and miniaturization. So we had to train them and to start the collaboration, but remotely. Hong Kong is 16 hours away from California as a time zone. So as Sumit was mentioning, yes, we were actually working day and night, and I was the person in contact mainly with Hong Kong. So I was the person who was staying awake at night to train them and to help them troubleshooting during the night or whatever was the issue coming up during the screen. And then once we ran the screen there, little by little, the virus became available in the US. So yes, we set up our BSL-3 thanks to the people of our health and biosafety department. And yes, since me and Laura Martin had already the training to work inside the BSL-3, well, we were the two people designated to work with the virus inside the room. So yeah, I mean, it was complicated at the beginning, but I think that we are more and more getting used to working there on a daily basis. For a long time. So at the beginning, I mean, Laura can give her opinion, of course. At the beginning, the toughest part, I think, is handling the respirator because, of course, it's not just, so the PPE is really complete. We have a complete respirator, 2 pairs of gloves. We get undressed, we redress with the scrubs, and because we want to be sure that there is no contact with our clothes or anything in ourselves with the virus inside, so we're sure that there will no risk of contamination. And so yeah, a tie bag, then the scrubs, the tie bag, and then the double pairs of gloves and the respirator. So at the beginning it was really tough handling the respirator, we were having shortness of breath every single time we were coming out from the BSL-3. I think that we're getting more and more used to that. I don't know if it's good or bad for us, but so we can stay longer actually now inside the BSL-3 if needed, although we are trying to minimize the time spent in there because it's not fun.
Like a spacewalk or something.
I'm sorry?
Spacewalk almost.
Yes,
Like
almost.
a spacewalk.
Yeah,
But it's important for people to realize, you know, the pressure and the technique and I wouldn't call it sacrifice exactly, but what you go through to help to bring us the possible cures and ameliorations. I mean, it's not nothing in that lab over there. It's a big deal, and you have to be super trained and skilled at it, like the three of you.
let me see if I can share a little bit of that. See if I can
You have that video?
here.
So Abel, just before you do that, I just wanted to give a shout out to our colleagues at Scripps Caliber who designed, had the foresight to design this library and share it with us, particularly Arnab Chatterjee over at Scripps. Really just been fantastic collaborators in the entire project and enabled us to be able to work with the folks in Hong Kong and get some of this work done.
Right, he's got a talk coming up on May 6th, right at Scripps. So
yeah.
Let me see if I can show you what that looks like. Can you guys see that? Is that the respirator you're talking about?
Yep.
Now, is this considered like BSL 2, 3?
BSL
Was this
3, biosafety level 3. So it's a negative pressure room. You have to gown in, gown out, like Laura was mentioning, and essentially you're trapped in that outfit for as long as you're there. I think 3 hours is your guys' record for being in there, right?
Wow.
3 hours and a half.
Does the suit start to reek after a while? Doesn't look like there's a lot of ventilation, but yeah. So this is one of the things, like people think of like outbreak in the big yellow suits and all that, and it's kind of sometimes that misinformation of how all these labs operate. But one of the things, my pet peeve is when they're doing these types of examples in the media. I'm going to show you here. Here's Sumit trying to handle stuff without gloves. See, that's a no-no. Walking around the street.
Hey, I thought we said no hard questions.
So, and then you've got, let me see here. You've got Laura trying to hand Sumit, what is this, a tube of the virus? And he's like,
where is this from? Oh, this is the Loa light.
It was just media, we promise, nothing infectious.
Yeah.
This gives the wrong impression, right?
Laura has gloves on there.
But she's trying to hand that to you. It looks like she's got a
Hollywood setup there.
Right? I love it. But let me go back here. Just, it's always nice to see how, show how what actually happens in a lab, right? Because a lot of people just will go by what they see on TV and the news, and it's not always accurate. So coming from you guys, I think it makes more sense. Let me see if I can get back.
And one of the big take-homes here is the idea, don't know if we stressed it, of drug repurposing that the lab is doing, that has come out at the top of the news, but it's as a possibly quick way to address COVID. But maybe you could address that right now. What's that mean to people, really?
Yeah, so the reason we decided to take this strategy, right, back in January, this is when the virus was largely contained to China, but the idea was that we saw this happening. It was a respiratory virus. It was a coronavirus. Coronaviruses are along with influenza viruses. The viruses are considered to have the highest pandemic potential because they're spread through respiratory droplets. So that really means that virus is a plane right away, right? It's a much easier virus to spread than, say, Ebola, which requires direct contact of bodily fluids, or Zika that you need a mosquito bite. So that alarm bells went off to a lot of virologists around the world, and so our calculus really was that, look, we need to do something and we need to do something quick. So typically it takes 10 to 17 years to build a drug from scratch, right? So obviously that was a non-starter. So what we thought to do is to take old drugs, right, and see if they happen to have serendipitous activity against the virus, right? And so what that allows you to do, because these drugs have already had a substantial amount of research put behind them, millions to $100 million of research to make sure that they're safe in people, we are able to leapfrog all of those regulatory steps that are so time consuming and money draining and say, okay, well, can we show that these drugs have any sort of ability to knock the virus down? Okay. And because we already know that they're safe, right, you can jump straight into clinical trials to see if they're effective. Now, remdesivir is a great poster child for this. It was an Ebola drug, didn't make it. People in the lab said, hey, you know, this also shows activity against coronavirus. Let's try it. And now we have what will be likely be the first approved drug against SARS-CoV-2, right? That was an educated guess because the protein in Ebola that remdesivir targets looks an awful lot like the protein in SARS-CoV-2. So people said, okay, well, let's try this. So that's what's called hypothesis-driven work. What we decided to do was leveraging this incredible library that the folks at Scripps built, where they amassed around 12,000 drugs, known drugs that have a track record of safety in humans, and asked, okay, which of those 12,000 might have activity against this virus, right? And so this is what the LARAs really were driving at, and now we're down to a set of 30 that look really promising, and then we're moving those into further studies in animal trials and then in human trials to see if we can't move the needle to add another arrow in our quiver in addition to remdesivir.
So the lay audience, how do you know what looks good to make that hypothesis? Is it a molecular? What that mean?
So we look directly at the virus, right? And so when you take virus and you add it to cells, that virus will make copies of itself. And there's various ways you can monitor, right, how well the virus is making copies of itself, right? And so we use different endpoints, right? PCR, I think some of your listeners will know what PCR is, but essentially it's the same assay that's currently used to diagnose the virus, right? It looks at the number of copies of the virus's genome or the genetic material of the virus, right? And usually when viruses infect cells, you get this exponential growth of the virus. And if any of these molecules, if you add it to that mix of virus and cells, knock down that growth, those are candidate molecules that are presumptive antivirals. And so that's what the kinds of techniques that we use. But this requires using a live virus, which is why the two LARAs have to get dressed up in the spacesuit. And testing 13,000 of these is no small feat. Abel used to work in my lab and really helped develop some of the methodologies we continue to use in that space. But it requires miniaturization and automation. That means that there are a lot of machines involved, and we take things that are usually 10 centimeters across and squash them down to reactions that can occur in the millimeter space, so we can look at 13,000 or 12,000 of these at a time. And so this is what Laura was on her iPhone in the middle of the night, trying to teach the folks in Hong Kong how to do it. And the folks in Hong Kong were fantastic learners, right? And yes. I never knew that you could do drug discovery by iPhone, but this is one of our lessons that we learned. And so, the other critical piece, and I want to let Laura Martin-Sancho jump in here, was, when we took delivery of the virus here, just getting the virus is not sufficient, right? You need to grow this virus up in sufficient quantities for us to be able to do these kinds of studies. And that was really no small feat. And Laura just burst out of the gates, and I think, in under 10 days, was able to figure out how to grow this virus in the lab and grow it to a point where we could run all of the assays that we needed to continue our studies. But Laura Martin, why don't you introduce yourself?
Hi, everybody. I'm Laura Martin. So I'm a postdoc in Sumit's lab. I think I started around 4 years now. It feels shorter, but somehow it is already 4 years. So yeah, my first contribution to the project was actually to start growing the virus. So we got it delivered from the BEI. So basically there was a traveler that flew from China to Washington, and the traveler was infected. So eventually they could isolate the virus from this patient, and then they have a repertoire for viruses here in the United States. So they expanded this virus, and this is the isolate that most of the laboratories across the country are using to investigate this particular virus. So I remember it arrived in the morning. It was the beginning of March, and then we said, well, the virus is here. Like, why don't you bring it in? And of course, we were very keen to start growing the virus. So because as soon as we could do so, then we could start validating the compounds that Laura and the people in Hong Kong started, and there were already some potentially very interesting compounds that we wanted to validate them here in-house. So I dress up. So as Laura Riva was mentioning before, we have to completely undress, and then we have scraps. And it basically looks like a spacesuit. I think Abel was showing some images. It is quite bulky. It gets quite hot in there. So the conditions, let's say, are far from ideal. The virus was, sorry, say again.
No, go ahead. Excuse me.
So the virus was shipped to us, and of course it comes with a lot of layers of security. It's contained under 4 different containment levels. So it was in a vial, inside of a bag, inside of a metallic screw cap box, inside of another box, inside of another box. So then there were multiple layers, but eventually at the end of it was just a very, very, very small aliquot. I actually hope that they will have sent a little bit more because the responsibility was huge. It meant that nothing could really go wrong because we didn't know if we cannot, or if we don't succeed at propagating the virus, we didn't know if we will get more of it actually. And we knew the time was ticking, so we had to really hurry up. So, I think it was a very interesting moment. Basically, it's the virus everybody fears, everybody's trying not to get infected with, and kind of our job was to propagate it so we can actually investigate compounds that can prevent us from getting very sick from this. So, I think it took around 10 days. I tested as many conditions I could possibly think of without using too much of the virus. And eventually I saw the cells were dying, and I knew that was the sign that the virus was propagating and that we came out with a successful protocol. So And
those are human cells that are dying, right?
So for propagation, we are using monkey cells. But so the virus eventually, what it will do, it will enter the cell, it will make a huge amount of copies of itself, usually from one particle that enters in a cell. Usually the virus can make 10,000 viral particles. So then basically the cells eventually, over the period of 48 hours, will end up dying from the infection itself. So then you have conditions where cells are non-infected and cells that are infected, and then by comparing just in the microscope, I could see that the virus was propagating and thereby killing the cells. So we knew that. Does this have
anything to do with the sort of what's talked about, that how fast it can take over a body? Like some people, it just kind of goes along for a while. You don't think it's that bad, and then all of a sudden, wham! and they have to be put on a ventilator or rushed to the hospital. Does that have, does what you three do explain something like that on a scientific level?
So we test in cells, right? We will not take, I mean, this is not in the context of the whole organism. So I think many people have to be put in the respirators because as the infection progresses, it can take over the lungs, and then eventually patients suffer from breathing. But so in when we propagate the virus, we have just a monoculture. So it will be only one cell type that we are expanding the virus on.
And I wanted to...
Sorry, just to jump in to follow up on what Laura said, there's two phases of infection, right? One is that you get the virus, you get it through your nose or mouth, travels down into your lungs, and then for most of us, if you do encounter your body, your immune system will clear the virus, and you felt like you had a cold. People who have to go to the hospital, right, have a reaction to the virus, which is called viral pneumonia, which is what really triggers the unfavorable outcomes, right? And this is essentially your body's immune system overreacting to the virus. And that is so in Laura's system, we don't have any immune cells in there. So we can model the virus growing, which is a critical component of disease, because without the virus, obviously you have no disease. But the other critical component is the way your body reacts to the virus. And for some people, for unknown reasons right now, they react to the virus almost too well, right? They basically have this over-exuberant immune response that ends up leading to a lot of other issues like lung damage and kidney damage and thrombosis and things like that. And so that is another way that people are now looking at treating COVID-19, is looking to treat that kind of inflammation that your body has in reaction to the virus that is really an uncontrolled response to viral infection.
Well, as a literary guy, I like that word, cytoclonal storm. It sounds
cytokine storm, yes. Cytokine storm, yes. That's what we're dealing with, right? And it's not a new phenomenon, right? So you see cytokine storm in diseases like sepsis. People are probably familiar with checkpoint inhibitors for treatment of cancers. Those are immunotherapies, which means that you're juicing the immune system. But sometimes when you juice the immune system, you juice it too well, right? And I think this is what's happening, is that your body's trying to respond. And sometimes for some people, it just overshoots, right? And so when you let the horses out of the barn, it's tough to get them back in. And so that's why people are dying.
Scary. Go ahead, Abe.
To just set expectations. Now, you said you had 30 hits from your last compound screen, right? How long? What's the time frame to get those 30 down to potential candidates that you take to clinical trials? Is it 6 months, 3 months, 5 years?
It's months, right? So the next step is animal model. So I got to tell you, it's the Wild West right now when it comes to these therapies and diagnostics, right? It's unclear what the regulatory guidance is going to be for any one of these, right? Hydroxychloroquine went into clinical trials without any sort of animal data, right? We won't necessarily need to get into why, but you can take your guess. Our philosophy is that now that remdesivir is available to patients, right? We don't want to give them another alternative unless we're sure that there's a reasonable chance that they'll do better or as well on this drug as they would have done on remdesivir, right? Because either you take this drug for the clinical trial or another drug. So we are really in favor of making sure that we have a data package in animals that suggests that these molecules will help, okay? And then after that. And those studies, not that long, 3 to 4 weeks, right? When we can move them through animals. Okay. Then we engaged the FDA. The clinical trial from remdesivir started in February, and we're talking, it's early May. So we're talking months, right? So this is, under what's called an EUA, emergency use authorization, right? So these are fast track. This is nowhere near what it usually takes to make a drug, but for obvious reasons, right? The regulatory folks are pushing these through. So there's a reasonable chance that if we are able to see efficacy that's comparable to remdesivir, or things that help in combination with remdesivir, that we could be getting into clinical trials and have some data by a second or third wave, which is expected to come in fall and then in the winter, if it does come, right? It's still not 100% clear that this is seasonal. If I had to bet in Vegas, and if Vegas was open, I would say it is seasonal. But again, jury's still out on that. We'll see.
So,
there's a Squawk part of this, and I'm a little bit bullish on Gilead. I had an annoying relative one time, and he thought he was going to die. He had hepatitis C, and he'd get up at 3 a.m, and all this sort of stuff. And when the trial started happening, I called his wife and said, "I'm going to save his life. You should go get this drug." Now, fortunately, he had serious insurance. We won't go into that. The drug cost $90,000, right. And the freaking guy's alive, still annoying me now more. But Gilead has, a hell of a pipeline. I mean,
they have from HIV to HCV to now this, right? Those guys are amazing. They're a powerhouse. They cured hepatitis C, right? And their HIV drugs, they have a preventative HIV drug that if you take it, the chances of you getting HIV diminishes to negligible amounts. Okay, this is what's called a pre-exposure prophylactic, right? That they are just doing incredible things in the antiviral space, and they're just a fantastic operation up there in Foster City.
Then there's other people. Well, I'd like to know what other companies maybe you guys think would have some promise, if we can talk about that. But also, there's the ethics of that. Gilead kind of rolled that out at a very high price, the hepatitis C thing, and people worry about the price of drugs and pharma and stuff. But my goodness, America seems to develop this stuff, and then the world kind of generics it. There's another side to how much it costs to create cures and pharma and all.
It does, right? And it costs about, we all like to hate on pharma, but pharma is going to be the people who develop a vaccine and develop cures, right? We in academics do the groundwork, but they are the ones who are going to take it across the finish line, right? Again, this is a philosophical and a scientific discussion, but Americans pay the brunt of the world's drug discovery bill, right? Because other countries, because they have socialized medicine, they are able to negotiate as a single payer and say, hey, listen, we're not going to pay this, right? But in America, it's what the market bears. And so it costs $1 billion to bring a drug to market. And there's an insanely high failure rate, right? It's a 99% failure rate. So, if you're betting $1 billion on anything, right, you want to have return and a high risk endeavor, right? This is not a 50-50 game, right? This is a high risk endeavor. So you want an ROI that's worth it if you're an investor, right? And unfortunately, the system is the system, right? And it's built on top of this capitalistic system where drug discovery and I think you can you can debate whether that's good or bad. I think competition really spurs innovation. So in many ways, I think it's good. But then, the issue of fairness comes into play, right? Do you charge $80,000 for something that will otherwise either have somebody need a liver transplant? The flip side of that is, liver transplants are expensive, right? More expensive than, $80,000. And, I mean, Gilead needs to make their next generation of drugs, right? They also need to reward their shareholders. So the shareholders will continue to invest in Gilead so they can make their next generation of drugs. So, it's a it's a more nuanced problem than, the simplistic, pharma companies are greedy, evil people, right? There is some of that, right? Every time you have profit motive, but it is a more difficult question than the pharma companies are just trying to gouge people, right? There are business considerations and high risks that these companies are taking to develop these drugs. And listen, if there's a better way to do it, I think we should present it, right? And I think, the US shouldn't bear the brunt of all of this, right? And part of that is because we don't have a single payer system where we can negotiate, but there are other ways to be able to negotiate and spread the hurt, right? But this is a conversation I think we need to have as a society and a global community. But I don't think there are any simple answers here. And I think that, just demonizing pharma is really not going to be the way that gets us to a fairer system that continues to spur the innovation that we need to bring cures to market as quickly as we're seeing with Gilead.
Well put.
Yeah, and I think like you said, it's complex and it's got multiple sides, and you got the other guys that say, hey, this is tax money coming out of our pockets that are going to NIH that are funding the research that's being done. So we are like investors. Why shouldn't we get an ROI on that? Why does that research go to pharma and then have them commercialize or capitalize on something that we invested in, right? So that's the flip side too.
It's true. It's, the academic model is really built on open sharing and dissemination of data, right? The whole idea is to put, for my lab, we took our screen and we put the data out so everybody could see it, right? Because we wanted anybody to take that data and find something that works, right? But I don't have shareholders, right? I don't have to answer to anybody, right? I can do that. I have the luxury of doing it. Right? Well, I'm sorry?
Just the two Lauras.
Yeah, just the two Lauras. Yeah, those are my two bosses. But other than that, as long as they're happy, right, I can do whatever I want.
Very cool. So let me see. So if this is my Joe Rogan moment and trying to simplify this for other viewers. So if going back to treatments and drugs and compounds versus antibodies, I'm trying to figure out a way to describe this to, say, a 6th grader to a 12th grader. And I was thinking, is a drug or a compound more like taking throwing a monkey wrench, throwing a wrench in a car engine's, while it's running, right? Or popping the tires or multiple, like you said, a cocktail. And is an antibody more like a tow truck, identifying certain license plates, going to find them specifically, and then carrying them away by a longer process, but more effective in eliminating the whole vehicle? Is that kind of a good analogy?
Yeah, I think that's a good analogy. I'm going to let one of the Lauras go ahead. I've been talking a lot.
I took auto shop. I forgot that.
So, Laura Riva, you're our drug discovery expert in the lab. Why don't you go ahead and field Abel's analogy there?
This is a very tough question, actually.
Sorry. It is tough to kind of translate science into layman's terms, right? It's taking like many years of studies and terms to like Joe Rogan it, like I said.
Yeah. Go ahead, Sumit.
I was going to say, Laura Martin, if you want to jump in, feel free.
I can go after.
Thank you.
I love Amanda's Schoolhouse Rock.
I don't know if I have to think about a war, for example. Definitely an antibody is the kind of how do you say that in English, actually?
what is it in Italian? And we can Google translate it.
Missile? Does it exist in
Oh
English?
yeah, missile.
Okay.
Yeah.
This is a good
So they
reason.
are the ones considered smart that are not smart, but that they target specifically. In general, they go and they just hit the specific target. So I would say that the antibody is more this kind of approach that goes and blocks the specific target, and eventually before. It's tough.
Yeah. So, but I think I like that analogy, right? The antibody is, it's a search and destroy, right?
Yeah.
The virus has a zip code, and the antibody is programmed to find that zip code, and
block
it
it.
seeks it out and blocks it, right?
Exactly.
Could it be like dating? Going from one to another till you find your match and you grab.
Yeah, exactly, right. It goes around as.
What kills
the antibody? I need my antibody.
There are these millions, thousands of antibodies running around in your bloodstream. I used to do a little bit of work for Biofluidica, and you could grab like a few circulating cancer cells out of millions, if not billions. I mean, it's a marvelous thing that you guys do. I don't want to sound, it's incredible.
It's biology, right? I mean, it's actually biology that developed this, right? We're just using it. I mean, they are exquisitely specific, right? I mean, these antibodies are just such an amazing feat of nature, right? To be able to discern the slightest differences between, different proteins and different viruses. So, yeah, so that I think that, Abel, that's a nice analogy, both the dating analogy and the car analogy on how antibodies work. They really, are able to specifically find and take out things. Whereas, yeah, go ahead.
Viral is it's like an obstacle. Like you're giving your body, you're taking an antiviral and you're putting an obstacle for the virus, whether it will be, assume it's a pathway that the virus needs to replicate, and then you are targeting this with an antiviral, so you make things very hard for the virus to replicate. So then when the virus enters the cells, encounters an obstacle, and then it cannot really propagate and make copies. So an antibody, as Laura was saying, is something more specific, like a missile, but an antiviral can be something like, I don't know, like if you're making things very tough for the virus to replicate, but it's not something, I mean, of course it can target directly the virus, but many times it targets something in our own body that the virus needs to replicate. So
it's like that fabric softener.
Question about zoonotic disease. Sorry.
Maybe it's like that fabric softener sheet you put in the dryer and eventually finds that spandex, and it's always like
and it clings on for dear life.
Yeah, you're like, how the hell did it find? It's always clinging onto that sock or that underwear, right? So anyway, sorry.
I'm out in rural Hawaii at the moment. We were noticing a gecko licking the guava juice off the edge of a glass. And so my wife got a little concerned about this. People are pretty worried about germs these days. I said, no. These are all coming from mammalian and bats. And then I thought, well, wait a second. Birds came out of geckos, out of lizards, right? A hummingbird is related to a Tyrannosaurus rex and all that stuff. But are these, I mean, a bit about what should we worry about? I mean, our president doesn't talk too much about MERS, which came from camels in some way, right? I mean,
so listen, there are viruses everywhere, right?
Yeah.
There are viruses in the ocean, there are viruses in plants. There's viruses that are considered to be of highest pandemic potential and highest the ability to jump species. But no virus comes from nowhere, right? Every virus comes from a reservoir of animals, right? So, Ebola is thought to have come from monkeys. SARS and SARS-2 come from bats. And as you mentioned, MERS from camels. So, what happens is that, just jumping between species is not going to be enough, right? It's very unlikely that, so don't stop, playing with animals, petting animals. I don't think that's, there's a lot that it's a very rare event that happens because the virus has to have some ability to already function in a human host, right? And so that is a really rare event, that there's no pressure for a virus in a bat to really grow in a bat and also be able to jump into a human host, right? So there's no selection for that. And so when it happens, it's just dumb luck that there happened to be some person interacting with a bat that just randomly happened to have a virus mutation that gives its ability to replicate in humans, right? But the second thing that has to happen, and this is probably no comfort to the person who's encountering the gecko, but the second thing that happens is that virus needs to be able to transmit from human to human, right? So that's the other obstacle. And that's not a small feat, right? So you guys probably have heard of bird flu, right? So bird flu is something that is out in China, infects a lot of chickens. It can infect humans, but what it's not good at doing, and it's pretty lethal, but what it's not good at doing is transmitting from human to human, okay? So those are the two things that are going to happen, and that need to happen in order for us to encounter a pandemic. And they're two very rare events. So the chances of you encountering one of these, more likely that you'll get hit by a meteorite walking down the street than you be the person that is going to be the patient zero, all of this. But, you know, you have these encounters enough, right? You start encroaching into wildlife areas where people haven't encroached before. You have these wet markets, right? You have factory farms where, you know, you have chickens being raised in high stress conditions that can transmit. You multiply that enough, right, times that a rare event actually becomes an eventuality, the more times that you set that possibility up. And so if you think back, right, you know, I mean, Abel, you're still young, but, you know, when, you know, in the 70s and 80s, right, I mean, you had one or two of these pandemics a decade, right? But if you think back, right, you know, we had SARS, then we had bird flu or swine flu, then we had MERS, then we had Zika, then we had Ebola, now we have this. And I'm talking the last 15 years, right? These things are happening more and more often, okay? And so, you know, I think that maybe we got a little bit complacent because we kept dodging these bullets, but we dodged these bullets because we were lucky, not because we were Superman, right? And I think, you know, this is not as bad as it could have been. It's bad. It's really terrible, but it's not as bad as it could have been. And I think, and I hope that if anything good comes out of this, that this is a wake-up call for us, both from an environmental perspective, the way we deal with our food, and from a scientific perspective, that boy, you know, we need to get ready, right? That this is not going to be a once in a century event. This is something that we need to prepare for over the coming decades and learn to mitigate all of the things that we did wrong that led to this current pandemic.
You know, it's you know, I think we lost Steve there. I don't know if it was connection. He's in Hawaii, so, you know.
Yeah, he's good.
Yeah. It's almost like it's better to have a more, and it sounds bad, I guess. I'm not sure if I'm going to come across awkward here, but it's almost like it's more beneficial to have a more lethal virus to pinpoint faster than to have something with but with a 14-day incubation. I mean, how many touch points do you have during those 14 days? Yeah,
it's a great point, Abel. You know, the thing is that this is 14 days, and a large portion of those 14 days, you're asymptomatic. So you don't even know you're sick.
Your eyes are peeing or whatnot, right? I
mean, So you know, you then, you know, this is this formula called R naught, right? It's essentially the number of people that get infected after a single person gets infected. And then the more you go beyond one, the more transmissible this is, right? Mm-hmm. But the more of an incubation time you have, the more chances you have of spreading it to people, right? And so if you're immediately lethal, right? Then I mean, it's terrible for the person who gets it, yeah, but it doesn't spread, right? It doesn't spread. And from the virus's perspective, it's actually not in its benefit to be lethal, right? And in almost, this virus has evolved almost to a perfect balance of, causing lethality. It causes lethality in a very small segment of the population, right? But the virus's job is to essentially go viral, right? And to make as many copies of itself in many people as possible, right? So killing the infected host too soon actually is counterproductive. So there's no selection pressure for the virus to be more lethal, right? In fact, a more successful virus from at least the virus's perspective would be something that is, asymptomatic for as long as possible, so it can spread itself around as this virus has done.
Like herpes, man. Sorry.
Please, guys. Please.
Welcome back, Steve.
To that point, Steve, and maybe the lawyers can speak to this, because there's a lot of misinformation about, was this a weapon? Was it released? How did it get out? So maybe 2 points here. What's the likelihood that this was, created as a weapon? And 2, what are the steps that you guys take to control the virus from, say, getting out of the Sanford Burnham, right? Because you're working with active virus. For people that don't understand those processes, what does that look like? So kind of 2 points there.
So I'm going to let the two Lauras who do work with the virus talk to you about the insanely stringent. So I'm not even allowed in the BSL-3. And it's my lab, okay? So
I've seen you on the bench. I wouldn't want you.
Yeah, that's true. Okay,
and that was 17 years ago.
Yeah, that's a fair point, Abel. It is. So listen, there are no absolutes here, but it is highly, highly, highly unlikely that this is a modified virus. Okay, it's too similar to what we see in bats for it to be modified, right? Was it released in a lab? I don't think it was intentionally released in a lab. Is there a slight possibility that someone in a lab working with the virus got infected and then went out and yes, I would say that it's improbable but not impossible. The Occam's razor is the simplest explanation, is usually the correct one, is that this came from bats in a wet market in Wuhan, China. But there needs to be investigation, right? That's I think that it's fairly irresponsible to make any conjecture otherwise until the data's in, and I think it's incumbent upon the Chinese authorities to allow a neutral investigation by the WHO to understand how this happened, right? But as far as precautions go, Laura Martin and Laura Riva, please talk about the really excruciating types of protection that you have. But in reality, there's a much higher likelihood that these guys are going to encounter the virus outside the lab than inside the lab. But Laura, you guys go in there on a daily basis. Talk about what you have to go through to get in and out of that, and also the training that you have to go through to even let your key card open that door into the. My key card doesn't work on that door. I tried it a couple times. So.
You want to go, Laura?
Up to you.
We can both answer the question, I guess.
It's the same. Okay. Let's say that whatever is exposed to the virus remains inside the room. So I think I would start from that point, and the fact that whatever is external surface of whatever we are wearing is removed inside the room, and we get into the anteroom where we remove our clothes without anything that got exposed to the virus inside. So the likelihood that the virus is transporting from the inside of the lab where we work outside is of course, we cannot say is equal to 0, but it's really, really unlikely. And concerning the training, I don't know, maybe I let Laura talk.
So I think we both, Laura, Riva, and I, we both had BSL-3 experience before. So then I think that we started the BSL-3 training. In my case, I had it from Germany. I worked with avian influenza, so I already had training in BSL-3 respiratory pathogens, and Laura Riva also had BSL-3 training before. So what we had to do for the Burnham BSL-3 training, we had a talk with Sylvie. Like we went through, I think it was a couple of hours, slides. We had to go through map of the room, all the procedures, SOPs, how to wear the PPE. Then we had another practical training. We even had this very cool thing. So it's called Microbe Glow. So then it's bright. Yeah, it was a lot of fun. Yeah, so then it will behave like basically you have it in suspension, so you can pipe it with it, and then you can aliquot it as if you were working normally with viruses in the lab, but it's luminescence. So eventually you need to finish your work, clean up as you will normally clean up after you are done, and then you can detect actually if you were clean or not. I mean, you can actually detect
it everywhere, right?
Yeah. That's probably why they don't let me do it. I feel that.
I just imagine that Sumit's face covered.
Yeah, I know. I look like Red Tide right now, right?
So then, I mean, we had like 3 sessions. There are some refreshers. So then both Laura and I, we went through that. So it was, I mean, eventually we get very well along with the safety people because we spend a lot of time with them. They have been key for the success of all this story. And yeah, so then I think only you, me, and safety, I think we have access.
And now Anshu, yeah.
Anshu, yeah.
Yeah.
So then it's really, really restricted.
Yeah.
I mean, as Sumit says, it's more likely that we go to the supermarket,
Yeah,
go around, and we get infected.
Actually, you're right. Based on just the number of gloves I see on the street and the masks, I'm more afraid of what I encounter on the street from other people than what I would see in a lab. So that's a great point. There's another thing, like we I think I mentioned this to Mitch Cronenberg, who was on the last show. We talk about how this use of hand sanitizers, right? And back in the day, antibacterial soaps were in, and eventually they moved them out of the market, or they're not as popular for. I mean, you guys can probably explain why better than I can. But are there any long-term effects, in your opinion, to overuse of hand sanitizers? Are we damaging our own microbiome? Are we removing the bacteria's innate ability to get rid of pathogens themselves by getting by killing them? I mean, what's your point there? Should we use them? Should we not?
I would say, Abel, right now you should. I think soap and water is probably the best thing you can do. But in the long run, it's kind of like when you have small children, right? You want people to be exposed to bugs, right? And good bacteria, bad bacteria. You need that microbiome, right? That is an essential function of your body, right? And so, the goal here is not to be Howie Mandel and become, afraid of everything. I don't know if the kids know who Howie Mandel is, but
dated yourself.
Yeah, I know, right? Yeah, but I mean, listen, exposure to pathogens is healthy, right? But we're in a situation where it's a pandemic, so I think short-term use is good. I think good hand hygiene is always a good idea, but you don't want to. I mean, listen, alcohol. So most of these hand sanitizers are alcohol-based, right? So they don't. So you're going to dry out your skin. You're going to lose your skin microbiome. All of those are going to have unintended consequences, right? And so, when things get back to normal, yes, wash your hands before you eat, but you don't have to be obsessive compulsive about it, right? I mean, it's okay to get sick, right? It's okay I mean, that's how your immune system stays on its toes. And so, just, continue some of the hand hygiene that you're doing now, but, you don't need to go overboard, right? I mean, we were just fine. Most everybody was doing okay with the number of times they washed their hands before this. And when we get over this, I think, again, if we go back to a little bit better hand hygiene, hey, maybe you won't get that flu infection or that RSV infection, but for the most part, I think, we're doing okay as far as hygiene goes.
How do we know that there's going to be a hiatus in the summer and it comes back in the fall? Are we going to kind of keep building numbers all the way through? You see all the people in different states going out, naturally, some of them, not so naturally others. Is there kind of a benchmark, or are you studying that, or other, people that doing that? Because that's kind of a big question out there.
Yeah, I mean, so we're not studying that, but others are. So there's, there's some predicted seasonality about this, just because other coronaviruses are seasonal, and also you can look to the southern hemisphere because it's their winter and they're seeing an uptick. But it's not a guarantee, right, that this will go away in the summer. It is known, it's been shown now definitively that the virus is less stable in heat and humidity. And
that's been shown.
That's been shown in laboratories, yes.
Okay.
And so, that's something. But how much that contributes to this diminishing in the summer is still unclear, right? We won't know but they are tracking these numbers. California numbers seem to be at least stabilizing, if not diminishing. And as far as at least most of the country goes, the original hotspots like New York and others, those numbers seem to be coming down. But you also have to remember that, a pandemic is actually very local, right? And so every county and every state is going to have their own kinetics, right? And so just because in aggregate you see California coming down, not every single place in California will follow the same trend, right? And so you really need to pay attention, in our case, to the San Diego County Health Services and take guidance from them, because, like the Bay Area had this virus much before we did, and they instituted quarantines and shutdowns before we did because it was a very local outbreak, right? And so, and when they relieve these kind of conditions, it should be done on a local level, right? And I think that this is what we're pivoting towards, right? So I heard Gavin Newsom today come on and say, look, counties are going to have the final say in what's best for them, right? The states will provide guidance and the federal government provides guidance. But, this is transmission, and outbreaks are going to be now local flare-ups, and they need to be contained locally. And you should be taking direction not from these broad national or international numbers, but what's really happening around you.
Because I was surprised at Orange County. I mean, there's a Sturm und Drang about can't go to the beaches or you can. Yep. But heck, that's 3 million people. Santa Ana is a real city, cheek to jowl. Right. And yet the number of cases and also the fatalities is very low.
Yeah. Yeah. And, again, it's going to come down to, how good your diagnostics are, how many people are getting diagnosed and quarantined, right? The sooner you can find people who are positive, quarantine them and everyone they've been in touch with, you're basically, I did an interview on KBBS, and they asked me to make an analogy, right? And the best analogy for us Southern Californians is wildfire season, right?
Yeah, that was good.
So when you have these brush fires, right, you want to figure out where they're breaking out and send in the planes, send in the fire trucks, and tamp that down before it jumps to 15 and starts burning down house after house. And that's exactly what we need to do with the virus, right? This will continue to be a slow burn, but when we have flare-ups, right, we need to go in with the cavalry and make sure that it's contained in the spot that it had, right? So let's say if I came out positive, me and everyone else that I had been in contact with in the last 2 weeks should shelter in place, and so that the whole community doesn't need to be in lockdown.
Because I'm thinking if I want to get out of quarantine, maybe, Hawaii's not such a bad place, learn to grow my own guava, up my surfing game here, I could come back in a few years. Once you have it figured out.
That sounds like a great plan. I might join you.
I guess you're saying that for the time being, though, I guess travel is going to be disrupted and just not going to be the same, huh?
I would say yes, right? I mean, I would not necessarily take my family on a plane right now, right? I think, for business travel, there's some things you got to do, right? And I think you'll see business travel coming back. If this abates in the summer, we may go to Hawaii or the Philippines on a family vacation. But right now, non-essential travel, I would recommend against it.
All right. Hey,
All right. So Steve, I see that you want us to wind up. So I want to leave off with the story that the two Lauras are going to kill me when I'm done telling. But I think it really greatly. She's already nodding her head.
Sounds good.
So we have this social distancing, and so especially Laura Martin is an extremely social person. She's Spanish, so she likes to hug and kiss people when they greet each other. And so we said no hugging, no hanging out in the lab. So these two, when they're in the BSL-3 in their protective gear, they're hugging each other because that's the only time that it's safe to hug somebody else that's not in your family. So to me, that was just a brilliant encapsulation of
we should have never told you.
I know you should have never told me, but
the Michelin women. Michelin women.
I know, it's such a beautiful story.
In our post debrief, maybe we'll invite you guys on Not in the Public, and we'll go through some of Sumit's karaoke videos.
Yes, we can do it. Fair. That's fair enough.
But hey, guys, I know we went over an hour. Definitely appreciate your time.
Yeah,
here.
My pleasure.
All the information. I know we can go on forever. It's a crazy complex topic, crazy complex time, and I appreciate the the explanations from high level to low. Steve, anything you want to add or?
No, it's fantastic. I think people listening, both the science folks got a real understanding of what's going on a little bit deeper, and those who didn't know, if they wanted to, learn how it's really done by some of our, high-tech heroes, really, which don't usually get to the media. So thank you. I was going to ask some stock questions, which ones you think might break out of the pack, but, from what are academics?
We'll do that offline, so offline. They don't drive off the prices.
Game show. But with that, guys, thanks a lot. I'm looking forward to eventually getting to see you guys again. It's been a while, Laura. I've yet to meet Laura Riva and Sumit, of course. Not the same, not having wings around.
I know, wings and beer, right?
Happy bench and small venue. But thank you very much.
Next week we focus on Baja California, I'm told by our chatter. And so that should be cool, from Bella Vista TV. You can go to Bella Vista TV on YouTube and subscribe. It's just like friending on Facebook, although this is going live. But once we get up to like 50,000, I think we're only up to 49,000, or maybe it's 490,000 views now. Don't worry with Fox or CNN or even NBC News. This is Bella Vista TV group.
TV,
Thank you.
Yes. All right, guys. Well,
okay, good. Stay well, everybody.
This is Science Squawk. We're out. Woo.